Hey everyone, so this is also another live stream. I'm doing a few more than normal. Usually they're actually like monthly or something like that with Aaron the grower. But today I'm going to be talking about various things related to the identification of microbes actually, and sort of a topic that I think a lot of people are very interested in, generally speaking, because I've been working a lot regarding a microarthropod identification in soil, and also I've been talking. I had an interesting conversation recently with Matt Powers, the permaculture student, um, who were talking about dark septate endophytes. And I think that led to an interesting topic because dark septate endophytes in general are actually rather um, they're not very well described actually, and they've been known for a long time. And I'm actually going to bring somebody online with me to talk about that because I am actually not. I do not understand all there is to know about the fungal taxonomy, for example, or bacterial taxonomy or something like this. So yeah, so hopefully when that individual, Growing Higher on Instagram, if you know who Growing Higher is. Okay, I see here. I'm gonna wait for the live stream to initialize. Okay, hello.
Sorry, I gotta turn that around. Hey, how's it going?
It's going good. They only do vertical
Oh shit.
orientation. Yeah.
Hold on.
So so hey there. Do you want to introduce yourself?
Hey, I'm Brandon Potter. I'm a Master's of Science in Biology. I work at New Breed Seed as a breeding technician. I also am a consultant with Mycophyte Solutions. We do like fungal plant interaction consulting mostly.
That's actually pretty dense, honestly. Like you said it very succinctly, but that's quite a pedigree, honestly, in my opinion. So you're
Thank you.
using a lot of this information very functionally, or a lot of your educational background, I should say.
Yeah, I'm trying to.
I know for a lot of people that isn't necessarily the case.
Yeah,
Um,
sure.
you shared with me a paper that actually, when I went to download uh, to my um, to my storage in the folder, I actually already had it. Apparently, I had downloaded it way before. This is a 2018 article called "High Level Classification of Fungi and a Tool for Evolutionary Ecological Analyses, not just analysis, but multiple analyses." Um, and I think that one of the most interesting things I'm curious to know more about is sort of like, because we both know fungal taxonomy is so complicated, it can be very difficult to sort of ascertain what is in your soil or on your plants, or even when it comes to like pathogens. I can't be sure, what fusarium strain I'm looking at, right? I can't be sure
Right.
of, or even like another one is when I talk about pest resistance, like with powdery mildew. I'm familiar with inoculation um, experiments, but when they do so, it's very critical that they know that they're dealing with a strain that's definitely virulent and has virulence traits and isn't like hypovirulent for whatever
Right.
reason. Mistakes like that can happen, and then you find that actually something is, or our samples for population. How would you put that? Like diversity?
Yes. Yeah, I mean, virulent. I could go into. There's like an interesting thing that was directly related to my research. I worked with Cryptic parasitica, and it has a virus that is the source of the hypervirulence, and they use a hypervirulent strain to try to biocontrol the fungus. Um,
That's
and
so
during
cool.
Yeah, during my research, I had to make sure the strains I was using to test, I was testing endophytes for antagonism against Cryphonectria, and I had to make sure the Cryphonectria isolates I had were virulent. So that's one of the this is like totally off topic, but one of the interesting side notes I found was that one of the isolates I got was from a forest that they had introduced hypovirulent isolates to try to biocontrol Cryphonectria. And I got a virulent isolate out of it, and it ended up being the most virulent of the characterized isolates. So it's kind of one of those things like it might have been introduced as a hypovirulent strain when it had the virus, and then it produced spores and lost the virus or whatever. And without the virus, it was actually like one of the worst pathogens. So it's one of those things you have to like, things are very complex, and like you might have an isolate that is, you might have a fusarium that's really pathogenic, or it might not be, and it might protect your plant against one that's pathogenic. So
I read about that. I just wanted to add. So like but I'm not very familiar with how people assess it, or I'm familiar enough to know that it's very difficult, and in some cases, maybe even a question that's like kind of impossible to answer functionally. You know what I mean?
right.
Or I don't know. Don't let me speak for you. You know about this way more than me. Is that true?
I don't know specifically, like it depends on the source of what's happening. I know there's like a Pseudomonas with it's like a SNP that's different, and that's the difference between the pathogen and
Just one snip. Wow.
yeah, the biocontrol agent. So it's like that's how fast it can change. You know, one gene can change an organism from a pathogen to a protector. So it's just hard to say, oh, you can do it with like, we're getting into other things, but like trying to look at an organism under a microscope and say, oh, that's definitely a pathogen, maybe. Even if it's in the group of organisms where there's known pathogens, it doesn't mean that specific isolate's a pathogen, unless it's causing disease in a plant. Or, you know, if it's linked to disease symptoms, then yes, that's a pathogen.
That certainly makes sense.
Yeah. So
I guess because I always like to bang on about the symbiosis continuum and how like for example, because of how we use it in common vernacular, if I say the word symbiosis, people will, I think, generally think of mutualism, but really parasitism is also symbiosis, commensalism is also symbiosis.
it's a continuum, and they can change, but depending the same organism, the same two organisms, if you're looking at a two organism system, can change from one being a mutualist to a parasite, depending on what the environment they're in is. So it depends on what benefits they're conferring in what environments. So it's, you know, we make these categories and we put biology, even species, into categories that aren't necessarily real. And we need to have this vocab to talk about them and to you know, work with them in agriculture or in ecology, whatever we're working in, we have to be able to talk about them. But that doesn't mean these are literal hard categories. Things move all the time. You know, the edges are blurred.
Yeah. As a person who looks at insects very much, arthropods in general, I was just, because I'm a massive paleo arthropod nerd, or I'm very close, or at least I'm very quickly becoming a good appreciator. It's just fascinating to see some of the work for like it was about like way back when like the chelicerate and the mandibulate lineages branched off. So what we got to like the pancrustaceans and the insects. And then we also got the mites and the arachnomorphs and the spiders and things. And like they were looking at like some antennal, like, you know, production, whether or not like it was synapomorphy or anapomorphy, whether, you know, with the rams. I'll put it in when I go to edit this video, I'll put in the paper, but I don't want to get too much off topic. I also do that a lot too. But it's just fascinating how much, like, I guess taxonomy is, we just have to make the ultimate concession that taxonomy is, any kind of taxonomy is arbitrary, kind of. It's just we're putting an order on a chaotic structure.
And things are changing. And I mean, I'm sure you're familiar with like the Claviceptacean endophytes, and they jump from insects to plants, and then they can be pathogens in plants, or they can be protective endophytes, and that's on the continuum. You know, like when they're pathogenic, they can abort the seed, but then if they're highly evolved with their host, like in epicloe grass endophytes, they don't make fruiting bodies anymore. They're just vertically transmitted. So it's this evolving continuum, and they came from insect pathogens, originally.
Yeah, and for those who are watching, like that would be like the Bouveria bassiana I love to talk about so much. I believe they are in that group, right? Or
Yeah,
well,
yeah,
cordyceps. Yeah, and
yeah. Cordyceps, Septaceae, the Mycorrhiza, a lot of the entomopathogenic fungi in biocontrol are Claviceptaceae.
yeah, and a lot of them are grass endophytes in particular. And I've heard about the epicloa endophytes that you're talking about. And it's just, it's just one of those things where, as over time, if it makes sense, maybe to make sense, it might not even. I mean, I don't like to put that sort of what's the word like for like that sort of influence. I'm trying to describe it in a way that doesn't like, I don't know, introduce uh, perspective bias.
Mhm.
You took the time. We were having a great discussion about sort of this topic about like fungal and microbial taxonomy and things like this and identification. Um, and you did up an outline that was really nice. I don't want to like put up an illusion that that's not the case, but the paper that you talked about that we started talking about like 10 minutes ago until I distracted us. Um, you brought it up because you thought that the systematics might be old, but like that it would still be useful for the general discussion. And what did you mean by that?
Yeah, okay. So like some of the more modern taxonomic papers for fungi. So fungi have historically been grouped into 5 phyla. And the more modern classifications have them up to 18. So it just becomes, and a lot of those phyla are like minor, like Microsporidia are really specialized. I mean, they're ubiquitous, but they're specialized intracellular parasites. Like most people aren't going to run across them in an agricultural field. So there's like a lot of those types of groups that are being elevated to phyla that, not that they're not interesting, not that those phyla aren't legitimate phyla, it's just a farmer doesn't necessarily need to know all about all of them. So some of the older classifications, like the 5 phyla system, is Basidiomycota, Ascomycota, Glomeromycota, Zygomycota, and I want to say Chytridiomycota was the 5th one in that system.
Is that the okay? I think I know what you're talking about with that last group.
I might have that. That might be outdated. I might be putting a different file in there than the one. But so those four Basidiomycota, Ascomycota, Glomeromycota, and Zygomycota. Those are like the ones that farmers are really going to have to, um, you know, deal with in their fields.
Mm-hmm.
So of course, Basidiomycota and Ascomycota are mushroom-producing fungi. So those are the ones that everyone's familiar with eating. They're the higher dikarya fungi. So they form dikaryon, which means they have 2 nuclei. They have sex. And then Glomeromycota are mycorrhizae. So those are the endomycorrhizae. Basidiomycota, Ascomycota also form ectomycorrhizae. So they form like the net around a root. They don't form the arbuscules inside of root cells in the same way that arbuscular mycorrhizae do. And then Zygomycota are really, Zygomycota isn't a phylum anymore. They split it into, I want to say, Zoopagomycota and I can't remember the other one. And it might be Entomopatho Mycota. Shit. Anyway, they split that one up. But it's basically the bread mold group. So it's like the hyaline, clear mycelia, usually without septate. It causes a lot of bread mold, usually a saprophyte. It doesn't usually cause disease, although there are some human mycoses and like post-harvest rots can be caused by it. So like soft rot on strawberry is usually Rhizopus, which is a zygomycete. And then of course, like one that people see a lot that is a fun one is dog shit fungus, Pilobolus, it's like a light sensitive, so it curls towards the light and then projectiles a little packet of spores. So that's one that's popular in like documentaries and science books and stuff, and is a fun lab experiment. Yeah, so that's not really that's just like a snippet of the modern taxonomy, but that's the major groups that farmers need to know about if they want to use fungi.
definitely familiar with, at the very least, the first three that you were talking about. What was the fourth one? Was the fourth one the zygo?
Yeah,
No, yeah, yeah.
Zygomycota,
Basidio, Asco, and Glomero. Specifically the Glomeromycota because of of course, because of the mycorrhizal interactions and the interesting research about how they could have been fundamental for perhaps like or at least they had a fundamental effect for like plant movement onto land. That's kind of interesting, or at least I've seen that in some research.
yeah,
Of
for
course,
Glomeromycota.
yeah. Are you familiar with some of that research as well? Like the prototaxides. People thought that it was maybe a plant, but maybe it wasn't. It was more of
Yeah,
a fungus. People are saying.
yeah. I'm not sure what about. I've heard of both of those things. The fungal symbiosis in early plants was probably essential for their own movement onto land. And I'm not really sure. I know prototaxides have these like huge fossils that people find, and I don't know much more about it other than that.
And
And they,
I'm definitely not a paleontologist, and I think even then, I'm not even comfortable saying to what level of speculation it really is. Um, but I just think that that kind of stuff's very fascinating. So I was kind of, and if that was true, then that means that they've had this relationship for a very long time, and sometimes it's been lost, and sometimes it's been enhanced, and that's symbiosis in general. Um, I was gonna add, and I'm gonna bogart just a little bit more of this conversation. I have one more question. You did mention fungal sex. Is it not true? I don't really understand the topic too much, but is it not true that sometimes I've seen it described as in like pluses and minuses instead of like outright male female?
yeah, yeah. I mean, fungal sex can be it gets complicated, but you can have heterophilic or homophilic types of fungi. So heterophilic need to have a different individual to mate with, and homophilic can have sex with themselves, basically.
I see.
So you can think of it like outcrossing plants versus selfing plants. Like a tomato can self itself. Uh,
And
versus,
but like, why do they use the plus and minus symbology? Do you know, or is that
as
just
opposed, as opposed to what?
oh,
I,
as opposed
I
to
think
like male, female,
it,
or sometimes Z in butterflies and things like that. Z chromosomes and or am I saying that
well,
right?
yeah, I mean
Yeah,
there aren't really male females in fungi. There's monokaryons and dicaryons. So they have there's a spore germinates and it has one type of nucleus from a haploid spore and it grows haploid. And then if it comes in contact with another fungus, it becomes a diploid. And once it's a diploid, then those nuclei don't fuse until it's ready to have sex. So it'll differentiate part of its body, like depending on what kind of, if it's an ascomycete, it makes like a female part and then a male part, and they have sex like that.
mm,
And that it could either do that as a dikaryon or two monokaryons that are coming together, and it does that, and then it forms a fruiting body, like where that happens. Um,
I see.
or
I
it can
see.
exist as a dikaryon, and yeah, it form a fruiting body. But those nuclei stay separate
Hmm.
until spore formation. So like in the fruiting body is where those nuclei undergo karyogamy, so the fusing of the nuclei together, and then they'll undergo meiosis and haploid um, spore production. So you can't really denote when it's a dikaryon, what sex is
Right.
it? It's both of them, and then the
Yeah.
haploid, what sex is it? It's one of them, you
So
know.
which ones? So that would be so maybe I'm speculating. So it's plus when it's when they're together and minus when they're separate.
I don't think so. I think usually I'm not sure which. I'd have to see like
Maybe I'm
what.
wrong. Maybe I'm describing something that's not even valid
No,
anymore. It's
I
possible.
do. I know like the plus minus. Like I've seen it in notation. I'd have to look at the specific notation. Usually, plus minus are like different mating types. So
Oh, okay. I see.
like
So I think I
plus might
totally
need a minus to
misunderstood.
be together. Hmm?
Sorry, I spoke over you. As I think I misunderstood fundamentally what the symbology represented. It sounds like to what you're saying.
Yeah, I'd have to see it specifically with what you're talking about. But usually when I've seen the plus minus, that's what it's talking about. Is a plus is one type of mating type, and a minus is another type of mating type. And they need to, like, a plus plus couldn't make a fruiting body. You need a plus minus, you know.
Yeah, that makes sense. I see. Well, so anyways, that was that was for me. I appreciate it. That was my bit of edification. That was a question that I've had in my head for a while, and so Wikipediaing it, I decided to ask you right now. Um, so anyways, I'll let you continue. We're talking about these orders being particularly important for farmers and things to be aware of, and they're very, very like the Mycota. I mean, that's everywhere pretty much. They're found globally.
Yes, yeah, and with most groups of plants, there's a few groups of plants people know, like brassicas. People generally recognize those don't form mycorrhizae for the most cases. I think alders, things like that, and they might form actinomycete associations. They're still forming microbe associations. There's just different ones, but mycorrhizae, endomycorrhizae are the dominant form of plant symbiosis for fruit. Yeah.
And I know that we actually talked about some things that might look like fungi but aren't. And I think the first thing that always comes to my mind is actinobacteria because they can make hyphae. So hyphae, for people who don't know, is not actually solely a fungal thing, right?
Right. It's basically just used to say like a filamentous body form. So actinomycetes make, or actinobacteria make hypha, and also Oomycota make hypha, which Oomycota are historically were thought to be fungi and are now known not to be fungi. Uh, so they produce both Oomycota and Actinobacteria. I keep calling it mycota. They don't form septa in the way that fungi do, and when they branch, they don't anastomose in the same way that fungi do. So when fungi come together, if they're compatible, they'll fuse, anastomize, and oomycetes and actinomycetes don't do that. They like branch, but they won't come back together and form a network. So that's kind of one way you could look at them in a microscope and see the difference. Although, again, like with both those groups, so Oomycota are generally indicative of like overly wet soils, but so you can kind of, if you have like a lot of Oomycota, you can say, oh, my soil's too wet, something like that. But beyond that, you can't necessarily say that Oomycota are a pathogen, especially for your crop. Like Pythium attacks cannabis and it causes damping off, especially in wet soils if people are direct seeding. But Phytophthora is another well-known oomycete that's a crop concern, is not known to affect cannabis. There's no known Phytophthora species, at least described so far, that has, especially the last few years, there's a new pathogen every couple of months.
I know, right?
But historically, hemp has actually been recommended as a biocontrol for Phytophthora infested soils.
I'd like to know a little bit more about that.
Yeah. So I'm kind of in Actinobacteria also. They're often responsible for disease suppressive soils. They can survive a higher temperature. So like when you're composting, you're usually killing a lot of your fungi, hot composting, and the Actinobacteria can survive in the pasteurized hot center. And then they can outcompete fungal pathogens. So that's one source of like disease suppressiveness is often the Actinobacteria in soils. And then you can inoculate soils with those Actinobacteria. They also, another benefit of Actinobacteria that people often don't recognize is they form symbioses with mycorrhizae. So there's mycorrhizae helper-bacteria, and I'm not really totally familiar with this interaction, but there's like a tripartite interaction there. So they're not antagonistic to mycorrhizae. Necessarily.
So it's a Actinobacteria mycorrhiza plant, sort of triple symbiosis that way.
Yeah, and I'd have to look into it more. I don't know if the mycorrhiza, if the Actinobacteria form hyphal interactions with the mycorrhizae or not.
That was going to be my next question: is do they interact like hypha to hypha?
Yeah,
Yeah.
I think they might. And Actinobacteria hypha are smaller. I'm not sure exactly how that interacts.
In my looking at the dark septate endophytes, I was looking at references for them, trying to find examples of like, like do we have like really a taxonomy for them? Do we have like maybe we have some ecological understandings, but it seems like they run the gamut. I almost feel like it's a distinction or a definition that lacks a lot of distinction, almost making it not useful as a designator. I don't know how true that is, but one of the interesting things you just reminded me of is that I was seeing some dark endophyte or dark septate endophyte to mycorrhiza sort of symbioses happening, which was kind of interesting, like integration with Hartig nets and things like that.
Mm-hmm. Yeah, I mean, it's a category. It's like the category endophytes. And endophytes can be separated into subcategories too, where some endophytes only occur in aerial tissue, some only occur in roots, some are vertically transmitted, some are horizontally transmitted. And there's like groupings based on that. And they're not taxonomic, they're paraphyletic. So a lot of things have evolved the same strategies. So
Right.
yeah, it's more of an ecological grouping than
Like a
functional
phylogenetic
grouping
grouping.
even. Yeah, they could be doing different things in there. They're just dark septate endophytes are all forming dark melanized cells inside roots.
But
So
is that really the kind of the baseline definition? Is that they just melanize?
pretty much they're
Hmm.
dark, they're septate, they're in roots. That's like
Yeah, I mean, hey, like I don't want to knock it or anything. Mycologists that are way more studied in this than I am not a mycologist at all,
uh,
so then pretty much all of them, right, are gonna know
yeah.
better than I. But I
And
just felt like it wasn't helpful for me when I was trying to find out like well, what is or isn't. But if it is simply just melanin production, I feel like a lot of fungi would fall into that then. So yeah, well, and
yeah,
also
and I
our
mean, I'm
endophytes,
sure
I
I'm
guess,
not a
right?
dark septate endophyte expert, so I'm sure a specialist in that would, those probably have subcategories where there's known,
Hmm.
groupings, ecological groupings within that. Because there's different mechanisms that can benefit plants, whether it's like sideriferous production for chelating nutrients or immune system interactions with the plant, where it's regulating the plant immune system. We don't. It can be different or both. We don't all know what they're doing. Usually, they're beneficial though.
Hmm.
It's the thinking. So
what was I gonna say? I'm not being very articulate. Um, you just reminded me of something, but it's gone from me now. Sorry about that.
no worries.
Yeah.
But
Mm.
well, one thing I wanted to say about mycorrhizae before we move on too much is the only type of mycorrhizae that form associations with cannabis are the endomycorrhizae, so the Mycota. So if someone's buying a mycorrhiza, most people recognize that now, I think. But if you're buying a mycorrhizal supplement and it has a bunch of Mycota spores or Sewellis, stuff like that, those aren't gonna. Maybe they'll do something in your soil and benefit you some other way, but they're not going to form a mycorrhiza with your cannabis plants.
That's good. That's really good information to know. And I am curious to know more. And I think that's something that I've talked about previously. And I've described it that like and I'm pretty sure this was the case, was that like if you don't know, like if we don't know the data, if we don't know, knowing how complex these sort of interactions are, I just feel like it's irresponsible for some people to say that something does or doesn't work, particularly without like, I guess you would need some sort of like sequencing confirmation at the very least. I know that's actually even a broadcast term. Like, what do I even mean when I say that? But like some sort of something that shows that like you're actually proving not just and even past that, not just interaction, like, but like, because what if it was parasitic? What was a
Right.
parasite we didn't even know, right? Like,
And I mean,
yeah.
you can do like root staining or whatever and look and just see that it's associating uh, But again, mycorrhizae can be parasitic if you're giving too much phosphorus, then it isn't benefiting the plant necessarily anymore. And we were going to talk about this later, but I know that you sent that paper about mycorrhizae virus susceptibility, and
Yes.
that gets into another topic, but I don't know if you want to save that. But
I mean, I don't mind talking about it here. Yeah. So that's I've posted about it before, so some people might be aware. But it's a mycorrhizal induced susceptibility, and this is specifically to viral agents. And I just think that that's just an interesting. I'm not sort of surprised considering talking about how complex it all is, but it does make sense because it does take resources to maintain these relationships, which is why they over time will like sometimes go away, or if a plant gets removed from one location and put into another location and then grown in a totally different parameter for a long time, then those delicate genetic. Oh, that's what I was going to ask you about. Um, Another thing that I've often talked about that maybe you can confirm or at least lend some sort of credence or discredence to is the nature of like how it is that much of these endophytes are modulating the immune system to what would be beneficial for the cultivator at the very least. And is it not true that just simply their foreign presence initiates sort of an immune response, or at least that might be, maybe they're doing maybe they're modulating upregulation or downregulation of genes and production of hormones and things like that. But like, I think I've also read that because they have a chitinous body and that sets off the effectors. Is there
yeah,
any truth to that, right?
I think that's so. Any endophyte that's getting inside a plant, it has to have some sort of antagonism to the plant to invade it in the first place and persist.
Hmm.
And I think that's kind of recognized as one of the universal effects of an endophyte is that it's going to cause an immune response that is just like a general upregulation of the immune response of the plant. Uh,
I think I feel like that really bears some salience, and I really appreciate the way that you put it. I I like to say I like to use the word colonize a lot. Now, I think people might even notice that in my posts and whenever I write things. I really don't like to use the word infect. I really like to use the word colonize because it's a little bit, I mean, I guess it's a little bit more neutral, I guess, a little bit. I'm just trying to show that the organism moves into this space
yeah. I
because I think, or what do you think about that?
No, I appreciate that. I kind of take the opposite. Uh, like especially when I'm talking about mycorrhizae, I like to say infect
Hmm.
to make people know that it's not necessarily like it could be negative.
I
they're.
like that. Yeah.
But
It
I see
sounds like
your
we
point
have the
as
same
well.
reasoning, though. That's
Yeah,
so funny. Yeah.
yeah.
But
So,
like, and that's the thing is that it could benefit, beneficial, detrimental is also context based. It's all context based. So, and I like that you're using infect in that way, in that sort of maybe sort of non-popular way. Like people don't realize, but these relationships cost resources, and sometimes they're highly costly, but good because they let
right,
the plant like survive, right? To some degree,
right.
sometimes.
Yeah, like a bad infection could become good.
Hmm.
It's just kind of I don't know. Uh, but one thing that led me towards the virus thing is and this is the other thing that I wanted to get your opinion on that paper that talked about uh, Rhizoctonia hosting cucumber mosaic virus, I think it was, and
Oh yes,
transferring
yeah.
into
He was talking about that. Yeah, yeah.
because those are Rhizoctonia and that virus also affect cannabis, so people might be interested in I know they used Rhizoctonia, which was a pathogen, but if this is happening and it's widespread, I know there was another researcher out of Australia that was looking at it was an armillaria clone that was introduced to South Africa, and so they like knew how old it was basically because it
Mhm.
was non-native, and so it's like a couple hundred-year-old armillaria clone, and they characterized all these viruses that had accumulated, like plant viruses. So if this is widespread phenomenon. I looked into like I have a plant virology book. It's probably 10 years old now. And the only fungal transmission of viruses it talked about was in Chytridiomycota, which is the cause of black scab of potato, I want to say. And it's also affecting frogs right now. So it's in the news for that. Um,
Oh, say the name again.
Chytridiomycota, I think the
Yeah,
one,
yeah, yeah. I've heard about the one that's affecting all the yeah, in like the tropical zones and specifically in like was it the Amazon?
I think it's pretty widespread, but yeah.
Okay.
Um,
Yeah. Dang. It's fun
but
to it's I mean it's incredibly terrific in the traditional sense of the word, really. That these sort of like trans kingdom interactions happen. But I guess people are just not aware of it. It happens all the time, we just don't know. But
yeah.
to get your but you asked my opinion. I
So yeah,
did say
go.
again.
Sorry. I mean, I'm curious if it is widespread in like hateful fungi in the soil, if they're hosting and maintaining plant viruses, what's the implication of that? And if they happen in mycorrhizae and things like that. That's that paper that talked about virus susceptibility. They proposed increased nutrients, like it was more uh, enticing to insect vectors because it had better nutrient content because of the mycorrhizal association. That was like one of their hypotheses. But I'm not sure if they looked at if the fungi themselves were the vectors and that like the fungi were just directly infecting plants, Like, is that the source of some of the viruses?
That's a good question, and I do feel like I think it's a safer way to put it that it's sort of underrepresented. That doesn't mean it's not happening more than we just happen to document. I liked that you brought that up because one of the cannabis viruses out there that I thought was really interesting was a cannabis cryptic virus, and the cannabis cryptic virus is a partitive and apparently it and Primula malacoides one, yeah, PMV1, yeah, Primula malacoides virus one, whatever. They're very closely related, and the interesting thing is that it's been speculated that PMV1 got into Primula because of a fungus potentially because they're very closely related to mycoviruses. I'm not explaining this very well. So up front, it's because they're related. They're very similar to mycoviruses, and so possibly maybe a fungal pathogen horizontally transmitted this virus or an ancestor to primula or an ancestor, right? And then it just never got out of the cell line, essentially, for however that worked out. And that possibly that's why cannabis cryptic virus is the way that it is, based on its sort of relatedness, based on the fact that it's not horizontally transmitted, it simply exists in the I'm forgetting the technical term now, but essentially in the cells. It doesn't it goes with when the cell divides, it divides with it essentially, and it's associated with hemp varieties, and so one would expect that cannabis plants that have some level of like yeah, like pheromone. There's a pheromone cultivar. There's a there's a few other ones. A lot of the really big names that are used in a lot of transcriptomics research. I see a lot of the same names like Finola and that kind of stuff. So it's just kind of it's fascinating to me. And I think after conversing with you about other like trans kingdom events and also just the nature of symbioses being so variable, I wouldn't be surprised if there's a lot more of that. I love looking at also some of the perhaps some of the origins of certain viruses come from escape. There's an escape hypothesis. I don't know how familiar you are with it about how certain lineages of viruses may have escaped from plasmids, perhaps because they share some of the same um, sort of mechanisms for interacting with cell biology. And I just thought that was kind of interesting. And so, like, there could be multiple like lineages or multiple ways that this could happen. And I think it sort of speaks to how like kind of like a ring species, um, another example is like genomes getting smaller in bacterial symbionts of aphids. You know, the free living microbe has genome size X, and then the symbiont that's very closely related, that's perhaps like a descent, like a parallel lineage, is like way smaller because it didn't have to have all of those different genes for traits that would be useful outside of the host, but now serve no purpose. So when they go away, it's no problem. Whereas other ones that help the host are still increased, but you know they become it's almost like you're cutting down a tool to be as simple and easy to work in a particular given scenario. I just
Right.
love that kind of stuff. I feel like it's all kind of related.
Yeah. An extreme example of that would be like mitochondrial DNA, where
Yes.
it's lost a lot of its genes, or they've moved to the nuclear genome.
I saw a depiction of it's like so much of our so much of energy on the planet. Oh God, that's going to sound a little bit woo. What I mean to say is that like so much of the interaction to the trophic energy, I guess I could say, is being transferred from like plants, right? Or autotrophs are responsible for that getting to all the heterotrophs is what I'm trying to articulate. But they're only able to do that because of the chloroplast, which itself comes from cyanobacteria. So really, is that cyanobacterial symbiont from long, 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 long ago doing all the
Right.
real work for all of us, hundreds of millions of years later.
Yeah, that's pretty amazing.
Yeah. Um, let's see. Well, there was another interesting topic. We're all over the place here. We were talking about identification earlier, and that was kind of the mainstay. When you are like sampling for fungi, how do you know? A sample is only going to help you so much, and you can't get a consensus. So how do you kind of evaluate that ecologically? I know that that can be very difficult.
Yeah, I mean, it's kind of a black box. I don't know how to answer it from a farmer level because like to really you can get a vague idea of where your biology is, I think. And if you look for main groups of organisms and you have diversity among all of them, I think that's like a good general look. But if you're trying to ID specific organisms, there's some tests that people use, PFLA tests, or I'm sorry, phospholipid fatty acid tests, PLFA tests, are kind of a standard and are pretty cheap. I think it's like $80 a test from Ward Labs. Uh, but even, okay, you're paying that for a sample, you need to take like a bunch of samples from your field, and then you need to do that on a regular time basis because the microbe populations vary based on season. Uh, it's just really variable. So I'm kind of skeptical about the use. I think they're good to do to get an idea of your soil over time, but to make specific recommendations based just on microbial populations, I think is rough. Uh, there's general recommendations a farmer can do that like will pretty much help regardless, like improving organic matter,
Mhm.
things like that. So
Yeah.
as far as
Well, a sample could be destroyed, right? Or not destroyed, but like broken or something. You had mentioned the Rhizoctonia having like some of them will grow at like angles and it's kind of unique and so you could tell that, but if it's shattered essentially, it's very difficult to
I,
really tell, right?
yeah, I mean, if people are doing like microscope analysis, I think it's very hard to tell what species you're looking at. There's some species that you can use. So let's talk about fungal idea, I guess. So when people idea fungus visually, usually you want to see the reproductive features. So if it's a fruiting body, you're going to look at the spore forming structure. So on like a basidiomycete, you're going to mount the gill and look at the basidia and how the spores look. There's various stains and things like that. You can look at features of the spores. And same for Ascomycota. You'll go to the lamellum and look at the spores, and you'll see spores and they form differently, but the characteristic of the ascus and also the characteristics of the spores, things like that. And then for asexual fungi, even more so the Canidia and Sporangiophores are important. So, if you have like in your refrigerator some moldy stuff and you see the green, that's usually trichoderma or penicillium forming spores. So if you can get a mount of that and look at the spores and the way they form the chains of spores, or however the characteristics of spore formation are how you identify the fungus. So if you're looking at soil samples, you might see some of that if you get lucky, but really you're just looking at hypha. And beyond, general groupings of fungi, you can't really tell much. So Basidiomycota they have characteristic clamp connections. So that's one that you could potentially look at and say, oh, that's definitely a Basidiomycota. If it's a dikaryon, if it's a monokaryon, it's not going to form those clamp connections, but it'll have regular septa. Ascomycetes usually have regular septa as well, but they have a different type of septa that allows their cytoplasm to flow, so they won't have necessarily two nuclei in each cell. They have more of a functionally cyanocytic body, which cyanocytic means like single cell. So something like a zygomycete usually doesn't have septa, so the cytoplasm just flows like back and forth more freely. So Glomeromycota also don't usually have septa. So, if you see fungi on your slide that don't have septa, they might be one of those two groups. Or they could be an Oomycota or an Actinobacteria, which are generally smaller in diameter, but it's so variable. The biggest Actinobacteria is probably the same size as the smallest zygo, depending on how they're growing and it's just variation.
Even some viruses are massive. The megaviruses, for example, and I think the Tupelo viruses or something like that was also kind of a recent designation. And then there's that famous example of the I think it's a fairy wasp that's like really tiny, but there's like an amoeba that's like this like as big as this tiny wasp, and it's just
Right.
it's a great example of how the diversity of like I guess that's just what's it called when you're looking at the sizing of things? Gosh, I'm forgetting the term for it, but it's like when you measure all the different heads of like a honeybee species to see what the kind of what the average is across species, because there's going to be some sort of difference. And man, I'm forgetting the
It's
name,
like
but
phrenology.
well, not phrenology, but well, it could be other parts of the body, but uh, teleomorph. No, that's a kind of fungal group. Anyways, well, actually, speaking of that, anamorphs, teleomorphs, that's still kind of a problem, right? Like different fungi can have very different, or the same fungus can have very different morphology just based on lifestyle. But I think we kind of already talked about that.
Yeah, I mean, so the teleomorph is the sexual, anamorph is the asexual. So you can have a mold and you have the anamorph form. And one thing that's funny about that is the anamorph form you can identify and it will have a different name than the sexual form of the exact same species. And it's just a historic hiccup, because usually the deuteromycetes, the ones that are common as molds, rarely form sexual fruiting bodies, or we didn't know that it was the same species, so we've given them two names. And because, usually in taxonomy, the sexual name takes precedence. In fungi with deuteromycetes, because the molds are so much more common and they are established in the literature, they've kept both names. So, like, what is it? Botrytis cinerea, I want to say is Botrytina fucaliana is
Yeah, I've heard of
the
that.
teleomorph,
Yeah,
which is the fun name. So,
yeah, that's right.
but it's the
Um,
same species,
it's like I mean, and I've heard that the ICN or the International Code of Taxonomy for I think it's fungi, algae, and something else. I think they also do chytrids too. Oh, was that the group you were talking about? The order was it Chytridae or
Uh,
Chytrida?
yeah, Chytridomycotas.
Yeah.
Yeah,
So I I
chytrids.
I think I've read that those are maybe actually what do you actually, that kind of reminds me. There are microbes out there also that are kind of like in this sort of nebulous taxonomic space, right? Like, and I think chytrids are one of those, right? Where they're like not fungi, they're like they're not fungi, they're not animals, they're not, you know what
So
I mean?
they're now grouped within fungi
Okay.
um, as their own phyla. They used to be grouped as a protist because so fungi are evolved. They're Pisciconta. So they're in our group where we have modal sporangia or modal gametes. So like our spore, I'm sorry, our sperm have tails and are modal.
Mhm.
Ancestral fungi are the same way. So Chytridium have mobile spores. So they were originally grouped with protists. But hyphal fungi and the higher groups of fungi have lost that, but they've evolved from organisms that have
Oh,
spores.
so the common ancestor had the motility. That makes sense.
Yeah, at least their gametes did. Yeah.
Okay. Right. Yeah. And Oomycota is the same way, right? Like, because they have multiple kinds of spores, and some of them are motile, and some of them are not, right? And there can be
Yes.
multiple motile versions, right?
Yeah,
Or am I-
I believe so. I believe both their gametes and diploid, they have modal forms of each.
that's pretty complicated.
Yeah, but they're actually so they're heteroconta. They're less related to us than fungi are. Um,
Hmm. Despite their similarities, that's kind of but it's phylogenetic, right? They're phylogenetically related. It's not just a nature of convergent evolution.
right. Phylogenetically, we're more related to fungi than the Oomycota.
But yeah. But I meant with the sorry, I changed topics. But with the chytrids though, chytrids and fungi are phylogenetically
Oh yeah,
related. Yeah.
chytrids are fungi.
Are fungi? Yeah, that's what I'm trying to
Yeah,
say.
yeah,
Yeah, because my
yeah.
because right because that was my historical understanding is that it was still kind of sort of ambiguous. But I really appreciate you bringing that up. And
Yeah,
that kind of
it's
stuff
very
is happening every
accepted.
year, right?
Yeah, um, it's
Are there
that's
other
why those newer papers, like I can't keep track of the 18 phyla.
right?
So
Who can? That's why we have a whole like multiple international organizations to try to
yeah.
deal with that mess. Are there other? I'm just curious since we're on the topic because like I feel like the words protist and protozoa get used interchangeably, but they're definitely not. Is that not true in your experience?
Um, I don't know. What do you tell me what
Well,
you're
isn't
thinking? I don't know.
yeah. So let me just make sure that I'm even talking about the right thing here. So like Protista is a group. It's not just like a it's not like
I mean, I think it's an
yeah
artificial group.
it's a art yeah. So it's a group, but it's not like a phylogenetically um it's paraphyletic,
Right. Yeah.
right?
It's just a convenient character group. But yeah.
But like
So
protozoan is that not like I'm showing my own lack of knowledge here, I suppose, in the moment. But you know what? Both of those are actually I think paraphyletic, are they not? Maybe that's the case. Yeah, I could be totally wrong here, but I've always felt like I thought that one of them. Maybe I'm thinking of a different P word, but regardless, do you have any other interesting things about certain groups that maybe have become part of fungi or bacteria but are not, like that just pop out in your mind? Because that's kind of where I was going with it.
I mean, Microsporidia are like a surprising group of fungi, which are
Okay.
intracellular parasites of animals. They're very reduced. Um, That's
I've heard of those with lady beetles. There's a microsporidian parasite of lady beetles that messes them all up,
yeah.
and that's
I've
why.
heard of some. There's some efforts to make biocontrol microsporidia for insect control.
Yeah,
So
I think I've heard some rummaging about that.
that's one way they might come and become relevant to agriculture more directly. Uh,
Well, okay. Let's see. Was there anything else you wanted to say about that? I guess with the identification and things like this, it's very difficult, essentially,
yeah, I mean, so another way you can, if you have specific isolates, you could grow them and make, things will form spores if you grow them over time in culture, and also characteristics of the culture, like the color, the speed, zonations, things like that can be used to identify fungi. But, you have to culture it to do that. And then,
you know,
of
I
course,
thought. Oh, sorry. You're the
no,
guest
go
here.
ahead.
I didn't do it. Well, I just wanted to maybe as you keep going, you might be able to. I'll inject this question too. Um, a friend of mine is talking about. I think essentially trying to like auto inoculate, not auto inoculate, but try to basically ferment or create a Beauveria bassiana sort of isolate that would be particularly pathogenic, or like trying to like produce it so that they can provide tons of conidia, actually conidia, or possibly even spores, and not just infective propagules, which are not really the same thing. And I'm curious to get your opinions about that. But like also, isn't that, I mean, my understanding is that's a very difficult thing to achieve. You have to have like um, a lot of technical equipment and staff to kind of do that. Because kind of what we were saying before about how virulence can be so um, context dependent and kind of change really easily. So you'd want to like, kind of make sure that those traits and those genes that represent those gene-based traits at least are being passed down. And especially like with Bouveria, you have to inoculate it on like living hosts, or at least like chitin having hosts, so that it has to
Yeah.
use those traits. Or what do you think about that?
So I mean, I don't know how he's selecting for pathogenicity in the insects. Do you know how he's deciding what?
I don't think they're selecting for it at all. Actually, they're just reproducing them, which is kind of my my concern.
Yeah. So I mean, if I were to do something like that, I would want to get the most pathogenic isolate I could find. So obviously, you're going to want it off of insects that are being killed by it originally or whatever. But fungi are notorious for breaking down, mutating whatever in culture. So you can have like a culture degrade. It's similar to people talk about it with plants, and with plants, it's usually an accumulation of viruses and things like that. And it might be the same thing in fungi, or it's just fungi are also, if you have a multinucleate fungi can not only form dicaryons, they can also uh, have vegetative compatibility groups where they aren't necessary. It has nothing to do with mating type, but they can still fuse. And then you have two different types of nuclei in the same fungi, which I'm not sure how big a concern that is into a pathogenic fungi like that, unless there's more than one fungi infecting a single insect, which probably happens. I don't know how that interacts. I forget where I'm going with this. Okay, so yeah, so like in culture, if you have a multinucleate fungi, one nucleus might be better adapted to grow in culture. So then that nucleus takes over the culture, and so
Oh,
that
wow.
in different ways the cultures can drift, whether it's mutation or not.
That's
So
interesting.
I would get that original pathogenic culture and make like a master culture and store it, and then always culture from the master culture. Because if you
Mhm.
just, if you like clone of a clone of a clone with fungi, you can lose your pathogenicity. There's actually a funny story about that. So culturing morels, um, people obviously tried to culture morels, and some people have been successful. There was a dude that had a patent on it in the 80s,
Oh,
and he had a system to culture morels, but it was culture dependent. And he was actually murdered, and his culture was lost. So people have not been able to uh, pull his system off to an economical scale. And they're also, there's lots of burn morels right now, so they're competing with that. But that's just one. The culture is important, so you can have these ideas, but if you let your culture degrade, your system won't work anymore.
Yeah,
So
no, that's kind of. Oh, sorry, keep going.
there's one more thing before I forget, because I was forgetting. As far as culturing these types of fungi, there's been a lot of work recently. There's a few people, I want to say terrestrial fungi on Instagram and mycosymbiotic on Instagram have done some work educating Americans on how to culture
Oh yeah,
cordyceps.
Mycosymbiote. I really like William Padilla Brown's work.
Yeah,
I also like a few of his songs. Very I like his style.
right.
Uh, yeah.
So
A shout out to him,
they, they've also
Mycosymbiote.
been culturing it in Asia. They're like miles ahead of us in Asia on fungal cultivation, but there's media that can cultivate cordyceps and other insect eating fungi. Usually they have like milk in them or eggshells or some combination of like a protein source.
Hmm.
And you can get them to fruit on it and produce sexual spores that way. Usually when people are mass producing spores for uh, inocula like that, I want to say they usually do like brown rice canidia production. And I know
Hmm.
that in mycorrhiza they do, hypha propagules. They count that as a propagule. I don't know that they. But usually, when I see like Bavaria products, it's spores.
Yeah, I've sometimes see them. I've seen labels depict it like canidia and infective propagules, and in my mind, I'm like, just give me the canidia,
Yeah,
you know.
I don't.
Which is why I
Obviously,
appreciate my friend talking about it, like trying to do that for like canidia, or like just straight give me the pure spores on contact, because I know spores on contact kills. I don't know anything about whether this hypha is going to get on and do its
right.
thing or not.
Obviously, it's going to have less of a shelf life than a spore. And yeah, spore. I mean, you'd have to have a big enough hunk of hypha to form like an aprosoria or to push into the insect, and a spore is like built to do that. I don't know.
Yeah.
I don't know what the research is for. I
Maybe if it got eaten, I could see like if like physically, like if a caterpillar or something, or some sort of chewing mouthpart insect, like maybe masticated the tissue and ingested it, that maybe that would be an effect. Maybe that would work out. But you're right, like purely from like a spore perspective, from like an actual infection, that does require, like what you say, an episorium and a penetration plug and all that kind of stuff.
Yeah, yeah. So I just don't know enough about if hypha of those fungi can do that.
Yeah. Yeah, I think that it'll be interesting. I'm curious, how do I want to say this? I'm very excited. Speaking of William and another other people who are working to make like genetic sequencing a lot easier for people, kind of at the individual level, at the like residential domestic level. I'm excited to see some of that technology become more democratized, and kind of in that sphere, I would like to see farmers and cultivators in general able to like kind of take control, or like have a little bit more of an ability to affect those sort of more nuanced things that right now we require like billions of dollars of infrastructure and that kind of a thing to research and articulate. It'll be interesting if that's even possible. But of course, that could also introduce other sorts of issues, I suppose, like people just not caring about biocontrols, or as much as I like Bouveria bassiana, I know it's found sort of globally, but it is a broad spectrum fungal um, infector, so there's ecological ramifications to that, I think. I don't know. What do you think about that?
Yeah, I, d- I don't know. It's just on that subject. If you're into mushrooms, Alan Rockefeller is really good with like democratization of PCR and mushroom identification.
Good to know.
And also B. Odin is a website, the ODIN, for like buying cheap refurbished PCR machines and stuff like that.
Thank you for mentioning that.
Yeah, the guy from that is it's an interesting guy. He also sells like home CRISPR kits and stuff like that. So yeah, as far as you're concerned, he's probably right up there.
I've heard
So
of some people doing some biohacking and some
yeah,
of the interesting. I think I saw a Batman Beyond episode about like splicers or something when I was like a teenager, and it's funny to see it. Like we're
happening.
here, we're
Yeah,
kind of at this like proto period where that very well could be a thing that people do. Nanomachines, son, and that kind of stuff. I don't
yeah.
know if you're a Metal Gear Solid fan. I certainly am. But
So I don't
uh,
know. As far as like GMO and environmental effects, you know, I I think it's like any tool. You can make tools good, you can make tools bad. If we want to make a killer virus, we could do that. If we want to, you know, make a plant that photosynthesizes a little better, we might be able to do that without having negative effects, you know. So should we rule that out? I don't know. Um,
yeah, yeah,
but
I go. I definitely feel like there are applications that might warrant it, whereas other ones might be a little trivial,
and
perhaps.
yeah, and yes, and it depends on the context. Like, what is the escape risk? There's people, even like with herbicide resistance, like in the escape risk, an herbicide resistant crop, that's if it escapes into weeds, that's a cost for the weed. So in the absence of herbicides, that's probably going to die out. You know, if it's
Mhm.
not being selected for, there's no reason for it to be producing, putting effort into being herbicide resistant. So like evolutionarily, I'm a little uh, things are more nuanced than like, oh, herbicide resistance is out in the wild now and we're fucked. Maybe not.
Right.
It's probably just going to die. You know, as long as we're not spraying herbicides everywhere, it's not. It's a cost, not an advantage.
Yeah, that's always, and that's kind of always been like one of my favorite examples of like where when people making like herbicide resistant like soybeans and things like that, it's like no, you're do it the other way. Do it like create a chemical compound that doesn't hurt your soybeans. Don't, just like spray wantonly this, chemical insect or herbicide or insecticide or whatever you're using. Um, like you said, like there's ways you could go about it, like maybe that would be a little bit more straightforward, or you could go about it a way that requires other people to, buy and get a bunch of other things that makes you more money. And I just feel
Yeah,
like
right.
the
That's
efficient move
usually
is probably not that. And you could
yeah.
achieve that using the same techniques, like, but what is your end game? And considering escape into the environment, I think is something I think that a holistic look at an integrated pest management strategy should consider. But like you're saying here, in some cases, like you can consider it all you want, but we don't have necessarily a solution or an answer currently. We don't really know. Um, but another cool thing would be like sterile insect technique. I'm a fan of that, for example, and that doesn't. So I guess sometimes they use radiation to sterilize like males of mosquitoes or things like that.
Instead
Um,
of well, I know they do GMO sterile males.
they
They're
do
doing that
that too,
instead
don't
of
they?
that. I
I
guess
think they
that'd be
do that
less
for screw.
controversial.
Do you think they do it for screw flies? I'm not
I
sure.
don't. I don't know the I was familiar with a mosquito malaria male. It was a genetic modified mosquito with sterile males, and they were releasing sterile males,
Hmm.
and it was
Yeah,
working very well. But that if they're using radiation to sterilize them, that would kind of remove the GMO concern.
a little bit, right?
Yeah.
Yeah, because they just have to mate once, so it's like, does it even make sense? In my opinion, it's a question of like, does it really make sense to use that or not? I guess you could use a gene drive, but you could also maybe get the same effect with just simple radiation. I don't know if one maybe is possibly even just more economical for the taxpayer if it's like a government or some sort of other organization, rather, just making it less costly in general. I guess
Yeah.
that really just depends on your tech tree, essentially.
Yeah.
But yeah, I wanted to get back to like I don't want to waste this great work that you did. Uh, oh, you know what? I was looking into some of the research reports that we were talking about with like I did a video recently. I talked to Aaron the grower about pH and EC and that sort of a thing with regards to like microbes and also like with insects and how they utilize or how they also use like the pH of their gut and like how like the physics of those sorts of interactions happen where they can kind of neutralize reactive oxidation reactive I'm sorry, yeah, redox reactions, reactive oxidation species and that kind of stuff. And so when cells like lyse and they create these superoxides and other sorts of kind of compounds as they break apart in a programmed cell death, that can damage some insects potentially. But they've also got like melanized mandibles, and they've got like various parts of their gut that are going to be different, sometimes different pHs and ECs that might neutralize those reactions or create an environment where they don't catalyze as easily, or like maybe the phenol compounds aren't going to be as effective or effective, right, at harming or interfering with things like nutrient absorption and that sort of a thing. So there's a lot of like really interesting interactions happening. But where I'm going with this is that of like what was it? Yeah, oxalic acid being something produced by various fungi, like we were talking about with the DSEs. Probably lots of fungi produce it, but whether or not that affecting the redox reactions is only something that like good organisms or bad organisms do. I think there was some controversy about that.
Right. I mean, so I've seen people claiming that oxalic acid crystals along the hypha are indicative of beneficial fungi, and I really don't know much information on it. I was trying to look up how to like visually how to identify this, and I came across a bunch of papers that talked about mycorrhizal mats releasing oxalic acid to help chelate nutrients, and it does do all this sort of thing. Um, as far as visually using it as an ID, I'm still unsure because I feel like it's more context dependent because oxalic acid is also a virulence factor for pathogens. So going back to my thesis related to that, one of the virulence factors of Cryphonectria is oxalic acid. So SUNY, the University of New York, has a transgenic American chestnut tree where they put a wheat oxalate oxidase gene into the American chestnut, and they linked it to a xylem promoter, so it's like only being expressed in the xylem.
Okay.
I think that part's correct. I have to check. But anyway, so it has this wheat oxalate oxidase gene, and it's breaking down oxalate, and that gives the tree complete immunity virtually to what is otherwise a devastating pathogen. So that's
Wow.
like where I am coming from oxalic acid. So to hear like, oh, oxalic acid means it's a good fungus, I don't know. I need more information. Like, is it this crystal form that means it's good? I really couldn't find information about it.
I found a report in 2008 that I wanted to share from the American Phytopathological Society that's entitled Oxalic Acid is an Elicitor of Plant Programmed Cell Death During Sclerotinia Sclerotiorum Disease Development, by Kyung-Soo Kim and a few others. And I was just reading the introduction when I was coming across it in preparation for the live stream, because I had heard something about that before too, and this is sort of, it was a convoluted intro, and I apologize for perhaps the semi misdirection there. The reason why I brought up like redox reactions and things with like insects and that kind of a thing is that basically it's kind of like the same thing with like redox reactions. People associate them with plants, or they associate them with like the good thing, but like really all kinds of organisms, all life forms are using these really basic physiological processes, and then they're built and they're added on, kind of like how venom in a lot of organisms develops. First, it's used for a lot of things like reptiles and insects and arthropods. It's kind of like saliva, and it has some like digestive capability, and then that gets like modified over a great period of time, and then suddenly you have certain kinds of venoms and toxins and things like that. And the very same is, I'm sure, true for fungi, especially when we've already talked about how much various fungi can like integrate with plants, and they have to, like you said, they have to antagonize, or they have to defeat the immune system of the plant to some capacity to allow itself to enter in. And in the
Yeah.
past, I've talked about it like a three-way, to use a sort of a cybersecurity term, but a three-way handshake kind of a thing, where like there's a genetic component of acceptability, but I guess that's not always necessarily true. It might be more like somebody, metaphorically barging into your house and like being like, "Here, have this stuff. I'm going to take your money," and then like leaves. Like it might be kind of like uh, I mean, how consensual is the interaction, you know?
Right.
And in
And
the introduction, they talk about this report. They mentioned that oxalic acid. What was this part here? I did not highlight it. That's too bad. Well, basically, they were saying that since oxalic or oxalate specifically contributes to pathogenic success of the fungus that they had mentioned earlier, it induces programmed cell death, particularly, and that was like what made it really useful, as a sort of for the pathogen. So, like people will talk about plants becoming resistant because they have like a high Brix level, or because they have various compounds that they produce that are maybe antifungal in this case. So like so of course it's going to be resistant. Sure, okay, that's possible, but how do you get resistant to like what is essentially proteinaceous acid, melting your cell wall, right? Melting through the cutin, melting through the cellulose, melting through the pectin, you know, dissolving those barriers at a molecular level. You know, it
you
doesn't.
make
But if what
oxalate
you're saying,
oxidase.
say again.
You make oxalate oxidase, apparently.
Like, apparently, yeah, exactly. Is that you
If
would
the
have,
cell
yeah,
wall. Yeah, it's not going to just happen.
right, exactly. And we do definitely have other. I'm sure it's a great example of other things that do happen, and resistance does exist. And so I'm not saying that resistance doesn't exist, but mainly that like um, I guess you could say pathogenesis in general is variable. And I think people just sign the people. I think the main problem is people look at it as a monolith when really it's not. Different strains of an organism will have different abilities for virulence, and different hosts or would-be hosts are going to have different levels of resistance,
Right.
right?
And in like the mycorrhizae that form these oxalate uh, crystals in hyphal mats, they're probably, I need to look into that more, but they're probably doing that away from the plant. They're probably not releasing huge amounts of oxalate right in the root. Or maybe they do during infection, and they use it for different purposes. Uh,
Hmm.
and that's totally possible, but just saying that it's there and that it's good, I don't know.
Yeah, well, I'm reminded of like the biotroph, necrotroph, and then hemibiotrope, thing where like biotrophs like powdery mildews have to interact with the living system. It's like they're like, to use a metaphor, they're like hacking the code, right? They're like they've got a sophisticated biological communication system, and they're using that to siphon out the resources and keep the cell alive and whatever.
Right.
Whereas a necrotroph doesn't care about this at all. It's just going to destroy everything and just sop up the nutrients that come with it. So there's no nuance at all. It's a, And then the
Yeah, and
hemibiotrope can move between both.
right. That's what I was gonna say. Yeah, like botrytis can act like that, where it is is not killing the tissue, and then suddenly it starts to, and then it's a disease.
Yeah, so it's kind of cheating, and I think maybe it makes us feel better to say that like that. So how many people do you think really know about this kind of stuff? Like most people don't, I feel like, and I'm not trying to say that anyone's like lacking in intelligence or experience or information in a bad way that makes them, it's not an insult to say it. I mean, even I don't really understand how these things are even happening, and I only know it because people did the research, and I happen to look at that because part of my profession. But I do feel like there's just sort of a fundamental perspective about maybe nature in general, like forests, for example. We think holistically, or yeah, when we think of like a forest totally, we think of like all of the organisms working in harmony sometimes, or like the sort of the picture book perspective that like if I plant all these plants together, they're not going to be antagonistic at all, even though like that plant evolved way before that plant's lineage did, and these things existed in totally different places, and they're actually antagonistic. But because they grow fine, and you fertilize them maybe, or maybe you even add microbes that facilitates it, like that first of all, that doesn't mean it would happen naturally. Second of all, none of the crop plants we're growing for the most part are natural. They've developed quite a bit in various ways. And then also, there's no guarantee that in a more or in a situation in an environment that's not the one you're growing in right now, it might not it might not be the case. Maybe one of them's stressed, the other one's not, and it overtakes the other one. Or any individual one could just have something wrong with it and it just dies. Like it's all capricious, I guess.
Yes, to some extent, yeah.
Yeah.
And changing dynamic, and things are affecting each other all the time. So
Yeah, no season's going to be the same, right?
yeah.
So
Um,
there is some level of like community identification, right? Like there is some like with the Rhizoctonia you mentioned, and there are some organisms that you can kind of tell, or at least you can be an idea of what maybe general taxonomic group they could be part of. You mentioned this earlier a bit with the different like kind of septate and whether they're hyaline or whether they're septate or aseptate. Was there
yeah,
anything? I think there was some more you wanted to talk about that potentially. There were some research reports possibly as references.
I'm not sure. What are we talking about here? I'm in the
Sorry
middle of
about it. You just had we had here. Uh, oh, did I add these? Maybe no. The Fleischbach video one about assessing soil biological characteristics, a comparison of bulk soil community DNA, PLFA.
okay. That was
Oh
that
yeah.
was kind of talking about, yeah, like so for soil sampling for community population for microbes anyway. Like for microscopic analysis, you can get a view of, especially for larger organisms, you can probably identify those much more easily. But for microbial analysis, like bacterial analysis, you're not going to really be able to meaningfully identify things just with a microscope. And this is making me think of something that I didn't mention for identifying fungi, especially like yeast forms of fungi. In culture, metabolic tests are often used. So you'll feed them different types of sugars and things like that, and you can differentiate species based on what they're able to metabolize. Um,
Oh yeah. I think I've heard about that.
and
Yeah.
this paper, actually, the assessing soil biological characteristics, a comparison of bulk soil community DNA, PLFA, and biolog analysis. The biolog plates are like micro well plates that they're built on this concept where they feed different food sources, and then syntrophic, or like it'll identify syntrophy. So one organism needs to metabolize the food source for the other organism to access it. So it kind
Mm-hmm.
of like does a little different, like a higher level metabolic test is what that one is.
That's
So
interesting.
basically, like the DNA, the fatty phospholipid fatty acid analysis, and that metabolic analysis are all kind of different ways to give you an idea of what's in your soil, and they are all inaccurate in different ways, but they're reproducible, within themselves. So we're getting like snapshots of what the soil community, the microbe community is with those. Um, and I think over time we're probably going to move towards DNA more, but there's still a lot of debate about what sequences of DNA will give you good, um, granularity on the species composition, uh, things like that. So, like with fungi, since I'm most familiar with fungi, ITS used to be the standard, 10, 15 years ago, people would use ITS to make phylogenies. And then we found out, oh, well, ITS isn't really that great at differentiating some closely related species. So now we need they'll do 6 genes and do a composite phylogeny, things like that. Because individual gene phylogenies don't necessarily agree. So.
Yeah, that's something. That's just yeah, it's not very helpful. I wish nature was a little more simple, right?
Yeah.
Actually,
So
oh, you go first.
no, the point being is just that I think that over time we're going to move to DNA analysis, and that the price will come down for that and things like that. But right now, I think PLFA is probably the cheapest and most accessible if people really want to know or have a microbe analysis done.
I see. You reminded me that I was reading a report about high throughput sequencing. In fact, in the cannabis virus chapter that I recently, well, I didn't publish, I put it through to get published, working with a colleague of mine. And I mentioned this report actually because it was so cool. And it was about their high throughput sequencing tomatoes. And they found 14, in the span of a decade, we found 14 new viruses through that sequencing technique alone. And they said part of it's because of the technology becoming more easily accessible and less costly. And so that made it just easier for people to use. And so, look at all the benefit of that. I don't know, there's got to be more viruses in various plants. We know that this must be the case. In fact, we know that there are tons of them that are just yet to be documented. We sequence them, but we don't really functionally understand them. It's just kind of like a barcode at the end of the day.
So I have a
Yeah.
question in your opinion. Okay, so we identify a bunch of viruses. If they're not causing symptoms, should we care as a farmer?
Okay. Oh yeah, good question. So I would say that to some degree, no. But I think it'd be interesting to find ones that might even be beneficial. So
Right.
from that angle, I could think that would be cool. And again, that's also a thing that we don't normally associate the two words together, virus and benefit. But like you were saying, well, the Curvularia thermotolerance virus that I mentioned before is really cool to me. And that's another tripartite or organization like you were talking about earlier. This is a virus and a fungus in a plant that lets the plant live in a very, very hot condition in the soil. And if the virus is not in the fungus in the plant, which is a salt grass, then the plant will not be able to survive. And I think it's like a 10 plus degree Celsius degree difference
Yeah.
in heat. It's pretty significant. And it's something to do with the virus affects the fungus's ability. I think they don't even have the full process figured out, but it's massively interesting. It affects the physiology of the plant, of course, and I think that it had to do with just the upregulation of certain. At the end of the day, it's a sort of the stress response. So it was able to sort of modulate that, and I guess it worked out for the plant. And so that kind of stuff may not be as rare as we think. And when I think of phylosymbiosis theory and hologenome theory, which I'm a big fan of, which I also know is contentious. I don't want to misrepresent anything here. It's kind of on the, all things that are kind of on the bleeding or cutting edge as far as massive theories about life are concerned. There's going to be some caveats. There's going to be some perhaps refinement that needs to happen. But the overall idea that microbial organisms are going to be maybe related through phylogeny at the very least kind of makes intuitive sort of sense, but I don't want to mistake that for a confirmation bias, essentially. But I think that we might find that these things are more true that or more common than we think, especially as our ability to sense them increases in sophistication.
Yeah.
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 Also, if we think of it long term, like, pathogens are beneficial in the same way, I mean, to some degree, as like other things are for like, honing that immune system didn't get sophisticated without an adversarial thing. And so maybe at the strategic level, I wonder, how much that will play a role in how much humans want to, I guess you could say, orchestrate that or coordinate with that. But I think that's a little bit much. It might not be possible.
I don't know. I
Do you
can't
know what I
speak
mean
to
though? Yeah, that's
it.
pretty like heady a little bit. Like, and that's kind of like controlling the weather. Like, even if we could, it introduces some ethical problems, perhaps. Let's put it that way.
Right.
It could be misused without even intending it, right?
Yeah. That's
Yeah,
not the reason not to do something necessarily, though. I don't know. Just do it cautiously.
right. You spent all this time asking if you could, but you didn't ask whether you should. One moment. So, were there any other topics that we prepared for that you wanted to say? Because you did do a lot of great. You did really help me with my own edification here, and I think it's just an interesting topic about sort of microbial identification and also just microbial interaction with plants. It's a topic I know a lot of people love to hear about more, and I'd love to talk with you more about it if you were interested. But this was a pretty good primer. I made us go around and around and around in different parts. We could have had a much more structured conversation, so I apologize for that. But I
Well,
was
I
so
did
excited.
it too. That's fine. But yeah, I'd love to talk more. I think we touched on everything I like mentioned. Yeah, I'd say like the main takeaway is to really identify fungi visually, you need to see reproductive structures generally, whether it's sexual or asexual, and that closely related fungi can be either beneficial or harmful, and it really depends on the environment, the specific interaction, the specific, you know, plant fungus that are interacting, the isolates, not just the species sometimes, you know. Uh, so
I mean, that's
that's
how I understand symbiosis to be generally. So it's really helpful to get your opinion, and also like I really appreciate you bringing up your own, even was it your thesis you said?
oh yeah, my graduate thesis was on American chestnut and Cryphonectria. So
Yeah, I have longtime viewers know that I have definitely talked about the chestnut blight before and how much that's like, it's just surprising to me when people say that like natural plants can be healthy or whatever and they will have no problems and it'll all wash out fine in the end for the organism. But like I guess that was kind of what I was trying to illustrate before. Like, the environment is actually very, very lethal. It's nothing but lethal, in
it
fact,
can,
and
yeah. And
you know,
I
it's very
I could
harsh.
talk like for hours on the American chestnut. And the American chestnut and chestnut blight is a model system, especially for biocontrol using a hypovirus. Um, like it worked in there's reason so the biocontrol with the hypovirus worked in Europe on the European chestnut, and it doesn't really work in the US, and there's like a bunch of reasons why. It's a really interesting system. So
Why is it? I'm just curious.
mainly it's vegetative compatibility. Haplotypes in the US are much more diverse. So the
Oh,
virus spreads by compatible fungi meeting, and then the virus spreads when it anastomizes. So it's like they merge, and then the virus spreads with the nuclei. And there's only one or a couple types in Europe, so the virus can spread through the population there really easily. In the US, there's four or more types of fungal mating types, and the diversity means that unless you get the same, not mating types, unless you get the same vegetative compatibility group, they don't merge. So the virus doesn't spread as easily through a population. And
Oh, I see. I see. I've seen some really interesting research about hypovirulent botrytis strains and how, and I've definitely thought, well, gee, this would be a great thing to be able to introduce and perhaps use to some
that,
degree.
yeah,
I'd
that's
be
very
happy
cool.
to sit. I see the your countenance has changed, and I'd be happy to send you the research that I've seen about it. I think it's really an interesting topic, and now you've got me thinking maybe this could be a biocontrol agent potentially.
Okay, that's one more thing to talk about before. So, especially, so botrytis probably can form endophytic relationships with cannabis. It hasn't been shown, but it does with other plants, and I think that it does.
I
Just
think
on
that it
a,
must. Yeah.
yeah. So it'd be really cool, like with something like that, you can introduce the hypervirulent botrytis to seeds or seedlings, and then it protects them. And so something that's also a concern with cannabis specifically is so a lot of endophytes of cannabis are human pathogen concerns or mycotoxin producers, like Aspergillus. There's either direct Aspergillus niger or direct Aspergillus flavus. They're either direct human pathogens or they make aflatoxins. And penicillin also makes a bunch of toxins. That's a common endophyte of cannabis. So if we have like botrytis can also make some human toxins of concern, but if you have these like isolates that don't cause disease and don't produce toxins, aren't human pathogens, right? If you can introduce those to plants to begin with, you limit or remove a lot of the uh, microbial contamination concern later. You could even limit post-harvest disease potentially. Maybe not botrytis, because even a non-virulent botrytis probably is going to eat the plant once it's dead.
Hmm.
But that's just one of the things I'm interested in is trying to introduce endophytes that aren't harmful as a way to get people to pass microbial testing. And that's not going to work if microbial testing is total yeast and mold counts. But if it's
Right.
PCR for known pathogens and mycotoxin testing, then it could. Uh, so
So if you essentially if you domesticate, and I use that word kind of poetically, I guess, but if you're able to like, kind of rend this hypovirulent strain out of the environment, make it so that not only does it not create mycotoxins that we don't care that we do care about, that we don't want to, you can essentially create it, put it into the plant, introduce it intentionally, and it can act like it's out competing the other botrytis, right? Is that the concept?
that's the idea. It's
Now,
and that's how a lot of
how would it not allow the other botrytis to also exist in the plant? How would that work, do you think?
that's it depends on how systemic it is. If it's like certain endophytes are spread throughout the plant, especially with trees, a lot of the endophytes are highly localized and acquired during the plant's lifetime, right?
Mhm, mhm.
So if it's systemic and it really spreads throughout most of the body of the plant, then it could just be working through physical exclusion, where something can't come in if it's already taken up. And food competition, physical exclusion. There could be direct antagonism, maybe not with the same species. I don't know. It depends if they're compatible, all sorts of things like that. Um, so mainly I'd say the primary mechanism would be simple physical occupation of the niche.
Wow. Yeah, that makes sense though. And I have a question since we're on the topic of that. Um, I've often been very skeptical. Like some people have talked about like washing, not botrytis, but well, I guess botrytis too, but mostly powdery mildew spores from like flower material and things like that. And but like obviously, just like with aspergillus and like bread mold, like the mycotoxins, if they exist, are already in the plant, right? So that you're not going to be able to like get rid of it just with washing. And my other question is, do you, I don't think there's any real confirmation of this, but do you happen to know and have any insight about whether and to what extent powdery mildews might produce mycotoxins
So
that would
okay,
be detrimental to people?
so I don't I am unaware of any mycotoxins of concern that are produced to humans that are produced by powdery mildews.
Yeah, me
So
too.
historically, I've said like the washing thing when people ask me about that. Um, you're going to remove a lot of spore load and physical mycelia, so it might help. And is that material dangerous to consume? Um, Probably not, as long as it really is maybe it's making a mycotoxin that we don't know of, but I mean, there's fungi in every cannabis sample that you're smoking, whether you see it or not, so like
That's a good point.
its presence absence as, and I don't know.
Yeah,
More
and if you're
likely,
smoking Bouveria bassiana, that you applied so that it would, physically exclude certain things or literally be poisonous to an insect that goes to like chomp on it, like it is kind of at some point I'm sure this is true for a lot of microbiologists in general, people who look at microbes, but like it's probably very much a different perspective about what's acceptable, what's tolerable, and like having that knowledge, I think, really changes your perspective, or I'm sure it has for yourself.
yeah, I mean, there's microbes everywhere, Like,
Yeah.
but what generally I think the main concern there is if someone's allergic to it, and you could be
Mhm.
allergic to literally anything, and that doesn't mean you can't have you could have a very serious response to it, and it could be the powdery mildew. So for some people, I think it could be a serious concern. For most people, it's probably, it obviously lowers the quality of the flower immensely, but it's
Mhm.
probably not a health concern.
Right. And I think that like, without sounding like a powdery mildew apologist or something like this, you're like, yeah, it's cool to like, you should totally not worry about this moldy stuff. Like, but despite that, like, yeah, it is there's a difference between something that's not desirable for cosmetic or even just like quality reasons, which is totally a justifiable, valid reason alone. But then, like, if you're really truly asking the question, is it harmful and how, you better have reason, you better have evidence for it. And I think that's kind of been the delineation point for me is that, like, on the one hand, it's a QC problem that people should not really accept as consumers. On the other hand, um, you know, harmful, probably not from like the perspective that I'm talking about. And that's usually where my mind goes is
Yeah,
mycotoxins, but that's why I wanted to ask you the question because I
yeah,
think that sounds like a totally valid answer.
and I know of none from powdery mildews. And if someone doesn't want to sell that moldy material, I think that extraction could still be a valid way to remediate it. And, that's extraction
I
had a
know
bad name.
it's like toilet to tap. It reminds me a lot of the
Right.
toilet to tap thing, right? Where it's like with a name like that, and when you think about it, ugh. But
Yeah.
like,
As long
but
as there aren't mycotoxins that they're, concentrating in that extract, then I think that's a
right.
valid thing too. I would be more concerned with, like, I would never smoke something that had aspergillus on it. If you see
Mhm.
aspergillus, throw that away. That is both mycotoxin producing potentially and directly pathogenic potentially. But there's
Quick,
also
oh, keep going.
one more thing. There was a case of Cryptococcus um, causing disease in humans that was linked to dispensary sold cannabis in California.
Oh wow.
So, I mean, you're not going to see Cryptococcus on your plant material. So,
Yeah, right. I mean, and when I was doing the dark septate endophyte research, or I was looking at that research, I should really be particular about how I say that when I'm talking to an actual researcher. Um, is that and that was Cryptococcus neoformans. The reference that Matt had used was certain DSCs were in the Chernobyl, for example. The infamous Cryptococcus neoformans is the one that I think of. There are others, but I never saw Cryptococcus being described as a dark septate endophyte in the literature. That being said, it does make melanin and it does interact with plants. So I think maybe it would be considered DSC potentially under that perspective. I don't know.
it's not filamentous, so it doesn't form
Oh,
septa.
oh,
So
I
I
guess not
wouldn't,
then.
under that definition, I wouldn't call it one. But
Oh, interesting. Okay, yeah. See, there you go. It's very that's kind of I am kind of at a loss about that. But what I was mainly getting at was that like yeah, it was really interesting to see research, and it's similar to the inter-kingdom interactions that we've talked about multiple at this point. But it was the plants were a host, or were they were a host for the Cryptococcus to complete its sexual life cycle, and then that becomes inocula for mammalians like people. And I also wanted to ask you, since you had mentioned Aspergillus infection and consumers not being should not consume it if they see it, how would they know? What's specific for Aspergillus? Just I think I have an idea, but I want to cover all the bases.
Yeah. So when you see a moldy bud, it's going to look similar to botrytis. Um, there's a couple Aspergillus that I see on buds frequently. Usually it's Aspergillus like terrace group, which has brown spores. So if ideally have a loop. They make their spores, which they're called Aspergillus. They make their spores on like this globular like ball thing on a piece of hypha, and then there's chains of conidia that come off of it.
Mm-hmm.
And it's named after, I want to say it's like this thing used in Christian like
A flail?
like ceremonies where they like dip it in water and like hit it at
Oh
you.
yeah,
And
yeah.
it's
That's
like
like it's like a flail kind of a thing.
yeah, and it's like
Something
this
like
club shaped thing. So it's named after that, where it has this club shaped and then chains of conidia that come off of it.
I see.
Um, as opposed to botrytis has kind of like a branched conidia with like grape-like clusters of conidia.
Mm-hmm.
And it's kind of hard to see with your eye. Botrytis is usually like a gray-brown color, whereas
Mm-hmm.
Aspergillus mycelia is usually white. Sometimes it's a little tan in the Aspergillus terrace, but often the tan is from the spores. So Aspergillus terrace has like brown spores with that grape-like structure. And
Uh-huh.
then Niger is much more easily identified. It has the same like grape cluster structure to the conidiophores, but the spores are black. So it's
Mm-hmm.
like white mycelia with dark black spores.
And colonization could happen of the bud sort of invisibly. I know that powdery mildew can basically colonize for like 120 plus hours of the plant without like really kind of seeing the hyphae like visually if you're inspecting your plants like as they're growing. Same sort of concern with Aspergillus.
I mean, yeah, most of the aspergilli are probably endophytes, and then either the ones causing mold are pathogenic ones that are coming in late, or it's an endophyte that's kind of becoming like a post-harvest disease in a ripe bud. So it might have grown after the bud was harvested, or if it's growing before that, it's just like, on the cusp of an beneficial endophyte to a pathogen. So
Mhm. Oh man, that's the worst because
yeah,
like that means that you basically can't. Well, in that context, this is a hot take, and feel free to answer it delicately because I think it is a delicate question. But like, do you feel like maybe controlled environment agriculture, maybe that's a reason why people should go that way for cannabis, but I do feel like there's environmental concerns for CEA,
yeah,
like so
I mean,
that's interesting.
I'm I like sun-grown stuff, and the idea,
Me
like
too.
environmentally, like agriculture, out should be should be outside, in my opinion. But as far as like medically producing things for immunocompromised people, maybe you could do it inside. And like, if you're doing it inside, you have to have like filtered air, you have to have UVC lights, and it's really hard to keep it completely microbe free. That's kind of why I like an idea, even in those environments, of maybe inoculating plants with some kind of non-harmful microbe to humans,
Mhm.
because then they're still getting the beneficial, you know, symbiosis the plant would get growth-wise, as well as protecting against any contaminants that do make it into those spaces. Uh, but there's also ways like, I think it's called e-beam, sterilization, or there's different like radiation or
Mm,
microwave ways to sterilize cannabis flower after it's harvested and finished. I think they all affect quality somewhat. Um, but if you're immunocompromised and you want to take that medicine in herbal form, that's better than getting contaminated flower. Uh, so for those markets, I think either or both of those avenues could provide, safe medicine for people with that concern. I'm also kind of ambivalent about it for most healthy people because it's an agricultural product. You're breathing in spores right now. Uh,
right.
what's the what's the level of concern for the normal person? You know?
How many people, how many people are there that are super hyper fixated on what I just said about CEA? Because, like you said, even getting controlled environment agriculture doesn't mean that you're going to not have it, for one. And for another, how many people are worried about this, but they don't even think about their indoor air quality, which I've read is way more important since people spend so much time in shelters that we call buildings. Um, like, and we have molds in our kitchens and our bathrooms that we're not even aware of inside of our walls in a lot of people's cases, far and away from whether or not it's the case in their agricultural product. And I just feel like, you know, put your money where your mouth is, sort of.
Yeah. Not that it's not, it can be a concern,
Oh yeah,
especially like a heavily contaminated flower. You could have a high spore load, um, which makes a difference in causing disease in some cases. But in general, I think, if you're not smoking obviously moldy flower, and if it is, powdery mildew is different than aspergillus. So mold is not mold necessarily. Not that I'm advocating selling or smoking
right,
moldy flower
right.
as an ideal. I'm just everything has a risk and it's a perspective.
Yes. What is your threat model? A question I have to ask people all the time because they might not have even considered it in the first place. And some things are definitely way worth more consideration than others. And I think, what always gets me is the cases where the answer is that we don't know, rather than like yes or no. And it's easy to say I don't know because there's a dearth of information and it's all inconclusive, or maybe we don't know, but most of the information seems to point this way or that way. Both situations can be misleading. You'll never know. You'll never have 100% of the information when you're making a decision, any decision you ever make in your life. So really, yeah,
I
that's the
mean,
ultimate concession, right?
I didn't know cryptococcus apparently is a relatively common endophyte of cannabis as well. So I didn't know that till a couple years ago. And then I saw the disease. I was like, okay, well, that's not a big deal. And then I saw the case where it caused disease. I was like, oh, okay, maybe I do need to worry about it.
When I was reading about it, yeah, I was like, Cryptococcus, I've heard of like as like a pathogen mostly. So I was really surprised to hear it described that way. And then but I hadn't really looked into the research much, and mostly it's in like human pathology research. But I'm looking it up, and I'm like, wow, this thing is nasty. This thing is really bad. And some strains are, it's kind of like MRSA, where like they're getting a lot more resistant and a lot more virulent, and certain antifungal agents aren't working at it in the same way that it has in the past. And that's kind of scary. That's kind of
Yeah.
scary.
There's also, I haven't seen it in cannabis, but there's Cryptococcus gattii is also on the West Coast on eucalyptus. It can occasionally cause disease too. So
Interesting.
just another one to be out there.
If you go like hug a eucalyptus, are you liable to get a transmitted disease?
Um, maybe. I mean, it's just in the air. It's
Have
like
you
Cryptococcus. How do you get it
have you ever
other than
heard of pyroaerobiota?
what?
It's
No.
a I don't see a lot of research on it. I just one time I heard this word, this really long word, right? I guess there were some like fungi and possibly other microbes were like they're able to like um I guess one way that they escape I'm not saying that they like what I'm not saying is that they resist like the massive heat tolerance of like fire. But like I guess during like forest fires and things like that, somehow what happens is that they get pushed up into the air with like the convection and the
I didn't
ash
know
and
that
all.
term, but yeah, I saw a study on that recently where, yeah,
Really?
dissemination of microbes from wildfire smoke. Yeah.
Wow. Yeah, and so yeah, that stuff is also really interesting. And for my Golovinomyces sicariserum pest primer video, I I saw that there was some interesting research showing like just how much like East Asia and like North America on like the Pacific Ocean, like it's very common for like powdery mildews apparently to like invade like North America to East Asia, and then they pass into like Central Asia, and then kind of vice versa across the ocean, not from human transport, but from wind,
It's
supposedly.
natural. That's crazy. Yeah.
Or like wheat blight. I've been following the head, and I'm forgetting the name of the head wheat blight or the head stem blight or whatever that's like ravaging like Africa, and like it's interesting to see the research where like they're showing like the different the various SNPs and the genetics showing their movement like up and down Africa and into Central Asia and then into Europe and then you know back around to like East Asia. Just, in the span, and then they're showing like how far the distance was in the span of time, and it's like they're moving like many thousands of kilometers in like 6 months or a year, and that was like in 2017 or 2018, I think. So I don't know. It's just I like to think of things strategically, even though I think we lack the capacity globally, at least, to really create a very competent, I guess, response system. But we're getting there, anyways. Oh,
Sorry.
wait.
Um, I got distracted. This comment that's been up there for a long time. I don't know how
Oh,
to. I don't know.
what's the comment?
Single fungus can either be sex minus or sex plus, and it takes both to reproduce. It depends on the mating type loci. I don't. I don't know about that.
Oh well, we could take a look at some comments too, actually. Actually, one of the worst things about Instagram Live, I think, is that it's actually they don't you can't save the comments with the video. I technically I understand why that doesn't work, but I kind of wish that you did get like a spread somehow if there was some way to do it because it'd be kind of cool to see people's interactions here represented. But you're saying, are there any other comments you wanted to comment about?
I don't. That's the only one I. Oh, I didn't see. I could scroll. I don't. Not really.
It's definitely not an intuitive system. They definitely didn't make it intuitive at all. It's very hard to scroll through. Phytoalchemist always has great comments. Plant pathology is an obscure science from a public POV. People don't think about plant diseases. I certainly didn't before I studied it. Yeah, and that's been my experience as an IPM specialist. Nobody wants to talk about it or even think about it. They're just going to be like, "I'll just grow my plants good, bro," and that's all I have to worry about. And I'm like, well, sometimes that is the case. Sometimes you can have a couple of years where it works out really well, but eventually it happens.
I don't know. He asked if they were moving, if mycology is moving away from anamorph to heliomorph. Not that I'm aware of.
I know that the ICN made a ruling or something where like they wanted to basically say like one fungus, one name. And I think kind of like what you were saying, like there's a ton of literature that sort of describes like the two different forms, but I think they are biasing towards one form or the other as like the official name. Um, Not that that's going to stop people who are familiar with one over the other, like Botrytis cinerea, for example. Um, yeah, the comments is very, very, very hard to scroll through, unfortunately.
But I think mostly touched on everything that I had in
I
my
think so
head for the
too.
time.
Yeah, I really appreciated it. Honestly, it was a really heady conversation, which is my favorite kind, about topics like this. I really appreciate you generally just coming on and having the discussion.
Yeah, thank
Uh,
you for having me.
yeah, of course. And I guess we'll cut it here. And if people have more questions, they are certainly able to comment on the IGTV when it posts. And I'm also going to be posting this on my YouTube channel. And I'll probably be adding some of the research reports we talked about, just so people have good references, put them in the description, things like that, so that they can kind of follow along. And yeah, well, thanks a lot, Brandon.
Cool. I hope to talk again soon.
Yeah, let's do it.
All right. Have a good one.
You too.
Don't know how to end this.
I'm going to end this.